Okay, test. Yes, okay, it works. Great. Yes, our next speaker is Professor Dr. Francois Deacon. He is an animal scientist whose research focuses on giraffe ecology, population health, and habitat use in African savannas. His work combines field research, GPS, telemetry, and ecological monitoring to understand how environmental variability influences giraffe behavior and conservation outcomes. Thank you so much for joining us.
Hello, I hope you can hear me.
Oh. Yes, we can hear you. Thank you so much for joining us.
Okay, let's hope everything works. Thank you for the opportunity. It's a real privilege. I'm not sure if you can see me or only the screen, but I'll fill it in wherever I can. So thanks, Ido. Thanks, Fabricio, for the opportunity. For me, it's a privilege to be sharing this and the story, what we do, the research we are busy with in South Africa and on the continent, the conservation efforts we're busy with, and obviously everything boils down to education. So I know I've got limited time, but bear with me because there's a lot of action and there's photos and videos added to it. So if you look at that, very few people actually focus on giraffe on the continent. So I want people to take note of what we are doing and how we can work together and collaborate on the projects we're busy with. So this, for instance, was a project we started with 17 years ago. I had to come up with a design with a GPS device to monitor animals in the wild and in the Kalahari, and other places followed after that on the continent. But that device opened a whole new world for us as a tool to monitor the animals. So I'm a rangeland scientist. So now we have this sort of information and data coming in, but what do we do with this information? So data in itself is just nothing if we don't convert that into information and eventually into publications and all that. So someone is running my slides, so I don't know who's running my slides or if it's automatic, but let's see if I can override that. So this is just a design of after a few weeks when the paint is off, and that's how it fits the animal very comfortably, and there's no harm done. And we've collected thousands and thousands of location data. And once you have that sort of information, this is on a very, very big reserve where you collect that information, you create hotspots from it, and then the hotspots between preferred and avoided areas, we can analyze the soil, the plant samples, we look down to the animal, and then we decipher the information back to what we can do in terms of management or education or conservation efforts to preserve the land better. So I think plenty of animals across the globe, especially on the continent, has been moved to areas that's non-suitable habitat. And if you know what island geobiography is, we are protecting small pieces of land, but it doesn't mean it's the right piece of land we are protecting. So first we need to look at the habitat, then we look at the animals and how the animals fit within that resource and with the availability of nutrients and minerals. So part of my team, obviously we're a big team, not just locally, but also from international institutions. There's a couple of Germans in these photos, if you look carefully, and the Touchings is there. Some of the other institutions and academia are taking part of it. But on any given day, we had a capture and a translocation this Monday. And on any given day, I've got between 40 and 60 people, scientists, veterinarians. I've got 8 veterinarians in the team taking part in these adventures. So yeah, it's not just the adventure. Everyone's got a specific task. And once we do it, we focus on the broader picture of how we collaboratively analyze the data and how it makes best sense. But there's a couple from Vienna, Austria, the Netherlands, Canada, Brazil, Peru. So in any of these pictures, you would maybe recognize some of the people, I hope. There's Jason Putual from Canada. He's helping us with the reproduction studies as well. yeah, so I could name you so many, but that's just the aerial view, just before we let the animals stand up and walk around freely again. Obviously, we want to convert all the information that we are gathering into scientific publications. So this was a study we did with the Royal Veterinary College from London, Christopher Basu and John Hutchinson, and we used the drones to monitor the animals' movement, and yeah, that was interesting. And then we also get access sometimes to unfortunate dead specimens that died for some or the other reason, and we make best use of that at the university as well. Anatomy, physiology, a lot of dissections, plenty of our students are working on that as well, where we try and figure out the animals from the inside. So that is a really good way also to understand the animals better. And we've got plenty, I've got 8 veterinarian students from Hungary and other places in the world that come and do their practicals and scripts with us at the university. And during this process, we also get to publish this information, the anastomosis, the valves, the heart blood pressure, everything that we look into. And eventually that, for us, is a big breakthrough, because we understand animals better. When we administer sedation medication or translocation, we can actually understand the animals better. There's just some of us doing some of the dissections in one of our dissection labs on campus, and then we code the different arteries and valves, and everything eventually turns back into publications. So just to be sound on this, we are being monitored all over. So we have our own paperwork, not just at the institution, not just provincially, not just nationally, but on a global scale. We need to stick to all the rules, all the ethics. So I think any of you that are doing science work or research work would know it doesn't just happen. You need to follow rules, and it takes months of planning to get to do that. This was just one of the DNA studies, the RNA studies that we did. We published that as well, just to see the relatedness between individuals, between different reserves. So we do a lot of genetic studies. The accelerometer data, if you could see that the device or the GPS harness behind me, you get a lot of accelerometer data, sleeping patterns, how the animals communicate. We're investigating that with audio morphs as well. Recently, we really effectively perfected the way how we collect semen samples from animals. So in our biobank, we've got thousands and thousands of baby giraffes waiting to be transferred to the rights of the recipients. So we're doing that with wild animals as well, and expanding the gene pool, where we can also sex the sperm between male and female sperm. And when we do the in vitro fertilizations on the oocytes, we can definitely develop embryos in vitro, and after 8 days we can do the transfers of these embryos as well. So that is what we are really focused on at the moment, the reproduction studies. There's just some of us in the field as well. We're working with various partners and people that has done this that is doing it for a living, like Absolute Genetics or RAMSEM, or some of the other very, very valuable institutions that not just work locally, but on an international scale as well. So there's us just doing some field work. A lot of this day we're collecting milk samples and all the biological measurements in any case. Yeah, so that's what we also did on Monday, and we were quite happy with our outcomes and what we achieved there. We're also doing some other experimental things, not just doing a typical castration, but we're also doing vasectomies. We published that as well. Yeah, I hope this video plays. Just tell me if it's not playing, but I hope this plays. No. Okay. So if the host can play this just after my talk, maybe it'll help. Or let's see if I can wait. Is it playing? This is quite a nice groundbreaking study we're doing at the moment. The focus area, the giraffe locomotion and the giraffe gait. Certain habitat, how the joints work, the muscle area, all of that, and what the strains and the possible constraints might be for the movement. It's the bigger picture we see in Africa is the giraffe numbers decline. My research also focuses on the spatial ecology of the giraffe, the areas they prefer, the areas they avoid, distances they walk, certain tree species they favor, and all of that contributes to our knowledge to manage these animals a little bit.
So that's what we're trying to do. We're trying to work out how their anatomy actually influences the way they get around the world. And we've come out here. It's a wonderful place to actually study these giraffes and to get some footage of them running to see how we can unwrap some of these mysteries. And at the same time, raise awareness for these animals, which are quite vulnerable. We're using our drones, first of all, to capture video footage, but we're also using some new technology on the drones to actually make a map of the terrain that they're running on. So a 3D map that actually gives us the elevation, gives us the texture, so we can work out, is it a rocky ground, what's the bushes, what's the vegetation like, and we can use that to work out some information from the actual video footage. And it's also useful to other people like ecologists to work out what the actual ground type is and how that interacts with the way the giraffes move. So it's very exciting to use this new technology.
Yeah, and like I said, we've published that as well, so you can look into that. And we're continuing with similar studies as well. We're doing a lot of high-tech stuff at the university, working with our physics department, microbiology department, chemistry, zoology. So we're publishing a lot of things, not just from the captive environment, but also from the wild, free-roaming individuals. We're using all the equipment to our availability as much as we have in Africa. We don't have as much as you have in the first world countries, but yeah, luckily we've got access to some. And then, zooming into the thermal regulation, working with Dr. Lisa Dodoni, some of you might know her as well, where we do some thermal high-tech information, not just from the outside, but from the inside, the animals as well, doing a lot of investigations on the x-rays and hoof care of animals, which you guys would be very familiar with, that we compared with the wild giraffe, like what are healthy hooves looking, and that we also recently published, that you can also go and source on the PLOS One. So looking at the hooves itself, that for us is an enormous breakthrough because it will really help your captive giraffe to thrive and be happy in the environments they live in. And then when we do get these specimens, we can do 4D imaging from latex and barium mixtures that we inject to the dead specimens, and then we can do these high-tech information gathering on how the split work and how the animal functions, its blood pressure, and where the splits go to. There's just a 4D image of one of the skulls that we have and that we're also working with. On a regular basis, we do that as well. And then the vet students do their projects on that. Recently, we've set up, last year, we've created a research center, which I would highly encourage any of you to come and see and join and help us, where we have a couple of animals there that we work on, that we regularly can do certain research projects on, and we test certain things. But for us, this is a breakthrough because this is really, really going to assist nature conservation. But our research will have a big boost. These are just the aerial shot from our structures. And I would love if anyone would come and see. And here's just a quick trailer of us running around in the field that you see how we catch the animals.
Not a lot about giraffe, a lot of questions to answer. You need knowledge to eventually turn this thing down if we can.
If we individuals, it is possible.
These colors could provide science necessary to save the rats.
This is in the end really going to assist nature conservation.
Yeah, I would encourage any of you to go and look a bit online of the work we've been doing. Again, we're not that visible like you would want us to be in Africa, but yeah, there's a few things. I'm fortunate to say I'm part of a big team just on our own academic institution at the University of the Free State. We have 64 people in 12 different departments already taking part in giraffe research or initiatives. And yeah, just students, postgrads. I've got 8 master's students at the moment, 4 PhDs, a postdoc. And in the last 10 years, we've published over 40 peer-reviewed publications that you can go and look into what we do, how we do things, and what we publish. So that's my story. I can honestly say with Monday's capture, we've reached our 300 successful captures, translocations, GPS device added to the animals. So we've done 300 successful captures up until now with this team. Very happy to share what we do, and I hope you've enjoyed my talk.
Thank you so much, Francois, for your talk. Yes, we are very happy to support your project with research on our giraffes here. I think that would be very, very great. And yeah, are there any questions? Yes, what?
Thanks, Francois. That was a great talk. Regarding the Ssmen collection on wild animals, is there any opportunity or any likelihood that they might be used for conservation breeding programs in zoos to improve genetic diversity of the stocks that we house under human care?
Absolutely, yeah. I wish I could see you, just to see you a bit more in person. It's so impersonal to be talking to a computer screen. But thanks for the question. Absolutely, I think that's the goal. Once we have perfected, I mean, in South Africa, we're fortunate to be working and experimenting our research on about 55 to 60,000 giraffe specimens. So I don't want to say we can afford to work on them, but it is basically easier than to work with isolated populations of very few. So the sperm collections obviously would need to end up in a larger, bigger goal and objective to get baby giraffes between subspecies as well. So part of our experiments is to use the South African animals as surrogate mothers. We do these embryo transfers into receptive females from different genetic pools and get baby giraffes coming from that. So if we can do that, we could not just do it locally, but we can do it basically in any captive environment, the zoo community or private or isolated population. So the experiment, we've done embryo transfers last year. Very happy with what we achieved with the entire process, getting to that point to actually do that. And part of our next step is to do that between the different subspecies and to see where that will lead. But it's a great question, because that would be the ultimate goal. It would be amazing if we collaborate with the Serengeti-Park, and we can send you some embryos from one or two of the different subspecies. I know you've got different subspecies in the park, and we transfer that to your individuals or your females. So and it would not just be for your animals, but it would be on a global scale. So the whole idea is to expand the genetic pool as much as we possibly can with all human intervention. But we've reached that point where we need to turn the extinction backwards and use all our available technologies. We've been doing it with livestock all over the world. So we can just as well do it now with wildlife, and we're doing it now with the giraffe.
Thank you very much for your response. Is there another question? Okay. So thank you very much, Francois, for your great talk. And yes, please join us if you can. And yes, we want to stay in touch with you, of course. We're looking forward for a meeting. Okay, then. Yes, our next speaker is Professor Dr. Bruno Baur, and he is a conservation biologist whose research focuses on biodiversity, species interactions, and ecosystem conservation. His work often examines biodiversity within human-managed landscapes, including zoos, and how these environments can contribute to conservation research and education. Thank you so much.
Good morning, everybody. I have two roles now. I'm speaking as a counsel of Basel Zoo, responsible for the animals, and I'm speaking as a researcher doing research in Basel Zoo. I will present you a study in which the native biodiversity between the enclosure in the Basel Zoo was examined. Basel Zoo is a city park completely surrounded by residential areas. It covers an area of 11.6 hectares. This is quite small compared with the safari park here. The zoo was founded in 1874, more than 150 years ago. And at that time, it was one of the first zoos in Europe. We have approximately 1.6 million visitors per year. We consider the zoo as a city park, as an oasis in the city. Our strategic planning has two basic requirements. First, the animal enclosure should mimic the habitat of the special species shown, and that means there are exotic trees, bushes, some rocks there. In contrast, the area between the enclosure should consist of native vegetation whenever possible. There are a few exceptions. There are some exotic trees. The intention of this is to promote the native biodiversity between the enclosure, and we would like to show this to the visitors too. Here's some illustrations showing you how it looks like. To test whether this strategy actually promotes the native biodiversity, we conducted a study that aimed to answer these questions. How many free-living native species are found between the enclosure of the zoo animals? Are there any rare and threatened species there actually? And are there unintentional introductions of exotic non-native species with the plants, with the trees? We used an all taxa biodiversity inventory approach. Actually, I managed to ask 45 specialists to help me in this project. And all the work, which lasted 3 years, was done on a voluntary basis, so they did not receive any salaries for this. They were coming from different institutions, including the university, the museum, Basel Zoo, the Entomological Society of Basel, and we also had some external experts from other universities. We divided the zoo in different sections, set or installed pitfall traps, other traps attracting flying insects, or light traps attracting night active invertebrates. We dug holes into the ground to study the earthworms, and we used different types of nets to sample insects on bushes and trees and flying in the air. To the results, how many species? Actually, we could determine to the species level 3,110 species of free-living fungi, plants, and animals. So they are for their own will there. They can go when they will, or they can come to us when they will. What does this 3,100 species actually mean? This is approximately 5% of the biodiversity in Switzerland on an area the size of about 15 football plains. We found, for example, 133 moss species, 91 lichen species, 45 springtail species, and so on. To give you a few examples, on this boulder we found 5 moss species. Or on this roof here we found 11 moss species. Here the summary of the findings, and please have a look to the column at the right side. We could not determine all the species, or we were not, or our sampling effort was not great enough to find all the species, so the expert actually estimated how many species there should be there. And you will see in some groups here in green, we encountered almost all species that may occur in the zoo area in Basel. But in other groups here in red, we did only find a few species. For example, the bacteria, we only found 8 cyanobacteria species. The other species we did not study. And in other groups, like the nematodes, we had no experts for nematodes, so we didn't know. So if we take the total of the species that may occur there, it's 5,500 species, approximately. It's 10% of the biodiversity in Switzerland. It's even more than 10% of the biodiversity in Germany, because Germany has less species than Switzerland. This is well known because we have the Mediterranean environments in Canton Ticino and the Canton Valle. You don't have these environments. So what does this mean if we compare it to zoo animals? We have about 650 zoo animal species, not individual species. So we have 8 to 10 times more species living in the zoo that are not labeled and are not recognized by most of the zoo visitors. We have also a considerable amount of threatened or endangered species. 113 species were listed on the red lists of Switzerland. What does this actually mean? A normal average nature reserve in Germany or in Switzerland has about 20 to 50 red-listed species. So the zoo area in Basel has more than double the number of threatened species. In other words, actually the zoo should have the status of a nature reserve. We have also new records for Switzerland, but this is not astonishing. This more indicates the relative moderate or even poor state of knowledge in some taxonomical groups. Now, this was a rather surprising result. We actually expected to have more exotic invertebrates, nematodes, or insects that come together with the exotic trees and bushes, but we only found 3 species. This is less than 0.1% of all the species. And to be honest, these species occur elsewhere in the city of Basel, so it's not proven that they were introduced in Basel, so they can come from other trees in Basel to our trees. So why are there so many species in a city park? There are different explanations. The first one is the historical development. This painting was made around 1850. It shows you the later zoo area, and we can recognize it on the hill there in the in the left background with this historical building. This is actually the zoo area with the river Birsig running from the right to the left here. And then about 25 years later, the zoo was founded. They used this area. And the next slide shows you the same forest as you can see here, 150 years later. It looks like this. And actually in this forest, we still found invertebrates that were associated to this riparian forest. So they could survive there for over 150 years in a city surrounded by residential area. There are other reasons. Another one is that the banks of this river actually allow the colonization of species. It's like a corridor running to the city. Sorry. Then the green areas, we are managing these areas in an organical way. So we use very little herbicides and insecticides. Then enclosures are renovated, they are changed. This creates new habitats. And finally, whenever possible, we provide additional breeding sites, nest boxes for birds, or these breeding sites for wild bees. When I summarize the study, we can say we have a really high diversity of native free-living species in the city park Basel Zoo. And a significant proportion of the species are threatened. But we have only very, very few unintentional introduction of exotic species. Now to the outreach. We did not have done this study for interest, and so we would like to tell our visitors what is actually occurring in a city park when you manage it in a biodiversity-friendly way. So we have guided tours focusing on native biodiversity, also for schools. And even in the normal guided tours for the zoo animals, we include elements of the native biodiversity. So native biodiversity is everywhere present. We have different measures to promote native biodiversity, such as piles of stones, piles of branches, sandy areas, and so on. And they are labeled, and we explain to visitors why we do have these piles of stone or these piles of branches. And then we have several demonstrations of native biodiversity. And one, the famous one, is the so-called zoo night. The zoo night is always the Saturday evening before summer vacation begins. And there we have a lot of tables and stands, and we show people native biodiversity. It can be bats, it can be other animals. And here the table or the stand of the Entomological Society, we have there a light trap. And we show insects and different things. And there are about 8 to 10,000 people attending this night. And we can explain to people very basic things. And I can tell you, I have learned that about 80% of the visitors believe that small flies, fruit flies, are young flies of the domestic fly. So insects grow. 80% of the people are believing this. They never thought about what's going on. So we have a lot of basic information to give to our visitors. And actually, I have the feeling that they really like it because they can discuss with us. You see, it's a distance of 0.5 meter. It's a hard work because this is 8 hours teaching, so to speak, but we like to do it. And of course, we also published the findings in peer-reviewed English journals, and we have also written a book about it. I have 2 books with me. I can give it to you free, if you wish. So we like to promote native biodiversity beside our zoo animals. Thank you for your attention.
Thank you so much. I think this is so important, and not only the educational area, but also the importance of zoos, or the role of zoos, I think. This is very, very great. So thank you so much, and I'm very interested in your book as well. Yes, are there any questions? Yes, okay.
I'm not sure if this is like your field of work, but it would be interesting. There's also like city parks in Basel, I guess, like regular parks, to compare which species are there, to see how much maybe more valid or more impactful a zoo is in preserving native biodiversity. Is there something planned like that?
Yeah, together with my research group, I'm now retired, but when I was still active as a professor, we studied private gardens and public parks in the city of Basel. And this is a small patches distributed over the whole area. And in combination, all together, they have a high value. But single gardens, they have a moderate value, but still, together with other gardens, they contribute to a higher biodiversity. Actually, this is not so well known, but cities can have a high biodiversity, and especially they can serve as secondary habitats for species that have lost their habitat in the wild. So this is important, and especially most people are thinking, what can I do in this biodiversity crisis and so on? And in their own garden, they can do something. Even if they have a balcony, they can have plants on the balcony, and they will see that pollinators appear there. So even with a pot of flower, you can contribute to the biodiversity of your environment. And this is the message we would like to give all to our visitors.
Okay, thank you so much. I can also repeat you. I think this is the best message for the future, and we would be happy if we could contribute to, for example, with a comparison from this zoo, because the Serengeti-Park is embedded in a more rural area, for example, and maybe this could be a nice comparison. Yeah. Thank you.
Okay, thank you very much.
The next speaker is Professor Dr. Theresa Götz. She is a researcher working at the intersection of artificial intelligence and animal behavior analysis. Her work focuses on using computational methods and machine learning to better understand animal interactions and behavioral patterns. These approaches help develop new tools for wildlife monitoring and conservation research. Thank you so much, Theresa.
Thank you. So today I want to talk about my elephant project first. So our big question was if AI can help us to to model the interaction of elephants. So I love math and animals, and I always try to combine both. Today I will talk about two studies. The first study is from the Limpopo region in South Africa. There we tagged, or my collaboration partners tagged 62 elephants, and we have GPS data from 11 years. We have almost 700,000 observations and also a lot of missing points, which is very challenging for the data analysis. At this study, I tried a social network inference algorithm, which I tried to identify the interaction based of movement residuals. In the second study, I had a look to Kasungu, it is in Malawi. There we have 20 tracked elephants over 19 months, and we have 4 hours prediction horizon and no missing data points. And this was a perfect data set for a graph neuronal network for interaction modeling. But let's start with the data pre-processing. So you all know GPS data. We have longitude and latitude, and here you see 2 elephants. But what my algorithm is seeing is not the landscape at all, only the absolute positions. And this is very difficult because I have a high variety in my data set. But I somehow want to combine these data. And for this reason, I used a principal component analysis to bring the 2 elephants in the same coordinate system. The drawback is that I'm completely losing any information about the surrounding, but my algorithm only has the GPS coordinates. So far, we not included any surrounding like rivers, roads, or climate informations. But with this pre-processing, I have less variation in my data set, and I'm able to use neural networks. Let's start with the first study in Limpopo. Here you can see all the tracks which were recorded. And I won't go into the deep mathematical details about this study. The basic idea is that we can model our movement with an intrinsic movement and a social motivated movement. This is the combination of both. The intrinsic movement I model with a small neural network, and then I calculate the residual, what was left, and this I tried to explain with a social interaction. I also did some statistical tests if the interaction is really significant. Here you can see my data set. So I have a lot of GPS points between 2013 and 16 roughly, and the other years I have less GPS coordinates. This is very challenging for analyzing data. So we see that I have a different number of elephants per year, and I calculated the significance interactions between the elephants. The quality is sometimes weak, but during 2012 and 2015 we have a strong quality of the data. This is my result. So I always give my partners, which are the ecological partners, some graphs. So you see each circle is an elephant, And if a line between two elephants is thicker, then we have a higher interaction. This is what came out from my model. The key ecological results in Limpopo was that we have a social coordination reduce the movement unpredictability. So we really have a benefit by modeling the social interaction. We have one cohesive matriarchal family unit every year, and we identified 2 females which are the leadership in this group of elephants. We have a lot of interaction between the elephants, but only 10% are significant. Then in my second study about Kasungu, there I had 20 elephants without any missing data point, and we have a sample distance between 2 points where we have 4 hours between these 2 points. So this was perfect to use a graphical neural network. The basis of a graphical neural network is also that I have the circus and the interaction, and through a neural network I try to identify the interaction between the elephants. What we can clearly see from the data, here you can see the, I always predict the next point of the elephant, and the precision is here roughly 2 kilometers. And you see some other methods. These are state-of-the-art where I want to compare to. You see the velocity persistent model and the persistence model. This is if the elephant stays at the same position, because elephants are not moving so much in 4 hours. And we see that our elephant graph neural network with the PCA as pre-processing is better than the baselines. And we see a history size of 3 is beneficial. History size of 3 means my neural networks knows all positions 12 hours in the past to predict 4 hours in the future. Also here I could identify for each elephant the strength of the interaction, if it was an outgoing or incoming influence, and if it is a high influence or a low influence. We have only one individuum with a low influence, and in this study in Kasungu, there were all the elephants without the last one were translocated into this area. So the translocated elephants were not interacting, or not so much interacting, with the only one which grew up there. We also identified the interaction over the time. We identify that if an animal has a high influence, it stays over time as a high influence animal, and also with the low influence animals. So our key findings were that the movement is socially driven. So we have a group trajectory history which captures real coordinate signals. We have, in this case, leadership is distributed. It was not a single elephant we could identify as a leader, so it were several individuals which draw the important decisions. And we had a stable social role around the year. What are our next steps? So we definitely want to integrate some environmental conditions into our model. For example, rainfall or river are very important things for the elephants, so we should definitely integrate it into our model. Also streets, houses, roads, they would improve our model to predict the elephant path. Yeah, we want to have a precise prediction of the next point from an elephant, and we also want to transfer this model to other species, like hyenas or jackals. We have also a lot of tracking data where we can apply my model. Now I want to use the remaining time of my talk to tell you about the Gaia initiative. It is a consortium of research institutes, nature conservation organizations, and companies. The goal is to create a high-tech early warning system of ecological changes and critical environmental events. This Gaia initiative is a big research project where we are trying to develop a new tracking device with satellite communication. I give you a moment to think about where this image was taken. I know the quality is not high, so it's not a high resolution image. But this picture is from Uganda, and it was taken from a vulture. So it was a camera which is placed on a vulture, and it was a wild vulture, and he was sitting in a palm and looking to the landscape, and exactly this recorded our camera. So with my team, I'm developing at the moment a new animal tracker with a camera. So the commercial tracker, you know, they have GPS, they have sometimes GSM, so mobile phone connections, also temperature and accelerometer data is common. But our tracker should also have a camera that we can take pictures. For example, for our vulture tag, it is that I can calculate based on the accelerometer data what the vulture is doing. So I know now he's flying, he's standing, or he's feeding. And if it is feeding, then I can take a picture. And at the moment we are sending this picture over GSM, if we have a connection, to us, that we can have a look what the vulture is feeding. So if you are interested in a collaboration, we would be happy if you want to test one of our prototypes. It would be also possible to transfer it to some other animals, but at the moment we are not so far that we can go undersea, so it should be on the land, okay? So, thank you for your attention. I'm happy to hear your questions.
Thank you. Yes, we are very interested. Concerning your question, yes, we are interested in a collaboration, and Ido told me that she has talked to you yesterday, and so, yeah, that's cool.
Great.
Are there any questions? Yes?
Yeah, first, thank you for explaining what I would say like quite complicated modeling and topic in such a simple way. I actually understood what you were talking about, which is great. I just wanted to say it was a very nice presentation and very interesting how you are actually studying the social behavior through GPS and coordinates. It's so counterintuitive at first, like to do it without actually observing the animals. But my question is, do you think this modeling can be used in conservation in a way to help to predict the movement of elephants to maybe reduce human-wildlife conflict or something like that? Thank you.
Thank you for the question. So for the data analysis, it's always the case that I'm getting the data without any knowledge. So I try to analyze the data and tell my partners what the data tells me. I have no idea about the ecological behind it. And afterwards he told me, these are the translocated and this one stayed. So this was really interesting, and I think this is the way how it should be done, not adapting the model till it tells what it should tell. Yeah, for the conservation, we try to estimate it more precise, and I have models where we can predict 2 hours in the future, roughly 600 meters. We hope that this would be one day enough to predict the conflicts between human and wildlife. Yeah, but this can be only done by the tracked animals, and there are a lot of without a tracker, so I think it will take some time. But this would be our big goal, yes.
Okay, thank you.
Exactly.
Thank you for the response. Is there any other question? Okay, I don't think so. Thank you.
Thank you.
Our next speaker is Kristýna Stehlíková. She studies wildlife, population dynamics, and conservation management. Her research explores how data from managed populations in zoos can help scientists better understand demographic patterns and conservation challenges faced by species in the wild. Thank you.
Okay.
Do you need one? Is it easier for you?
It would be better, but I
have one, but I'm not sure if we can connect it. So.
no.
Yes. Okay.
So hello everyone, and thanks for the introduction. So today I would like to share with you what I'm currently focusing on, which is what can managed populations tell us about factors influencing the population dynamics in the wild. Where we are taking the western giant eland as a model species. So firstly, you will surely agree with me that monitoring endangered species can be often challenging, as they very often are we have small populations which are fragmented and under low densities, and that's also connected with very often with low detectability. And simply getting long-term data sets which would be robust is quite hard. But on the other hand, conservation of such species required good understanding of the key demographic processes to be effective. So here we believe that the managed populations actually provide unique opportunity to understand the processes and demographic constraints in the wild, because in the managed populations we have detailed monitoring which is based on a long-term scope. And one of the animals which is like good for studying this is actually the western giant eland, which was once widespread across Western Africa, but nowadays we can find them only in Senegal. The last wild population is present in the Niokolo-Koba National Park, and for a long period of time this population has a low density and very restricted distribution, and together with its elusiveness, the monitoring of this population is quite challenging. But luckily, we also have the semi-captive population, which was founded in the 2000 by 6 individuals taken from the Niokolo-Koba. And then in 2008, it was separated into 2 management units due to some management reasons. And up to date, it presents one of the most detailed demographic data sets available for any antelope in Africa. We have really 25 years of detailed monitoring, and what is also important, this population has minimal predation and poaching pressure, and it has stable resource availability as it is supplementary fed during the most harsh parts of the year. So our aim was to evaluate how the wild and semi-captive populations perform under contrasting ecological and management conditions to identify the limiting factors for the wild population recovery. Short look at what the data we are using. So for the wild population, we have images from camera traps, and we use them to individually identify the animals and look for recaptures. And as I said, in the semi-captive population, we have very detailed stag-boo data based on direct observations. And for both of the populations, we also have some genetic samples, and with different approaches we were able to analyze for both of the populations, the population structure and size, breeding and survival rate, and also the genetic structure. So first, let's have a look at the different population trajectories. In the wild population, we can observe really slow population growth over the time and low density. When we look at the latest estimates, the density is just around 0.1 individual per square kilometer. And even though the population size estimates may indicate some growth, we have small data sets and quite wide confidence intervals, so there is not proven any significant difference in the estimates. Then when we look at the semi-captive population, it has a stable growth since its establishment. In 2004, the population had almost 200 individuals. There were not registered any population crashes over the time, and the population growth rate is mostly positive. So here, basically, the managed population reveals the potential for the giant elands for population growth under some favorable conditions. Then when we look at the age structure, here the wild population has significantly higher number of juveniles compared to the semi-captive. And in the semi-captive population, we have it's mainly adult dominated. Then when we look also on the sex ratio, in the wild, it's female biased, which is something quite what we expected for a polygynous ungulate. On the other hand, in the semi-captive population, the sex ratio is either even or even male biased, which is probably effect of the lack of some external factors pushing on the population. Moving to the reproductive output, here we would expect that it would be the biggest problem for the wild population, but the opposite is actually true, and the breeding rates in the wild actually exceed the long-term means of the semi-captive populations, and you can see that the estimates are in general higher than the annual breeding rates in the reserves, which is very positive. Then when we look at the survival, so for the females, it's quite comparable between the systems. In males, in the wild, the survival is a bit lower, but considering the ecology and social system of the animals, it is also something we expected. But the biggest difference we can see in juveniles, where in wild the survival was estimated less than 50%, which on one hand is quite normal for large ungulates in the wild. But you can see that in the reserves, the survival of juveniles is very stable, and also in general around 90% or higher. So here the managed populations basically reveals a much higher potential for the early survival than is currently expressed in the wild. Also a short look at the genetic variability. So here we can see substantial overlap between the two systems regarding the variability, which is positive in terms of some future management and potential reintroductions. But we have quite low overall allelic diversity across the populations. But despite that, we register no evidence of a heterozygote deficit and no evidence of inbreeding depression, which is basically telling us that the population is most probably not limited by the genetic factors, but some outer factors. So to somehow sum it up for you, when we look at the semi-captive population, it seems that it's approaching some demographic equilibrium. We have high density of animals in fenced reserves with very limited space for dispersion. We have high proportion of adult males in the population, which is putting them in quite high social pressures, and it's increasing the competition. Also, when we take the number of calves which are born and the number of adult females in the population, it seems that the reproductive potential is not fully expressed. And overall, it seems that the population is reaching some density-dependent regulation. On the other hand, when we look at the wild population, it seems to be well below its ecological potential. We have low density, high fecundity, and relatively high adult survival, which are all predispositions for the population growth. On the other hand, we identified several limiting factors which are probably suppressing the population growth. Firstly, even though there is high breeding, the recruitment into the reproductive age is quite low, which is probably driven mainly by predation and resource competition pressing on the young age categories. We have also quite low number of adult males in the population, which is probably caused by selective poaching, especially in the past. Nowadays there are more strict anti-poaching activities implemented in the park, so we hope that in a few years we will see positive outcomes of this. And lastly, the population is still limited into very small area of the park, and it seems it's a combination of both anthropogenic and ecological factors connected to some habitat connectivity, livestock encroachment into the park, and also human presence, as giant elands were proven to be quite avoiding the humans. But when we see what is going on in the wild population and contrast it to how the semi-captive population is doing, we can see that there is a substantial potential for the population recovery of the wild population, which is very positive. And that's it for me. So at the end, I would just like to thank all of our partners which are collecting the data in the field, to Antwerp Conservation NGO and my faculty for funding, and of course to all of my colleagues which are taking part in this research. And thank you for your attention.
Is the microphone going? Okay, great. Yes, are there any questions? No question? Okay, I think we have the whole day for further questions, sure, or the next day, so thank you.
Our next speaker is Paweł Borecki, and he works in a zoological conservation and reproductive biology. His research focuses on cryopreservation techniques and assisted reproduction technologies that support the conservation of endangered species such as the African penguin. Thank you.
Hello everyone, thank you for the introduction, and today I would like to tell you a little bit about the challenges in the cryopreservation of African penguin simians and the outcomes that we got despite of them. Why the African penguins? They are the only ones from the 18 species of penguins that are critically endangered. And they are predicted that by 2035 their wild population will go extinct. mainly due to the human activities, obviously. So what we are trying to do, we would like to preserve their genetic diversity before the wild population goes extinct. And to do so, we've came up with the multi-level approach project. First, we focus on the reproductive behavior analysis. Then we move to semen collection and the analysis. Then moving on to the cryopreservation. And at the end, we are making the comprehensive handbook that will explain step by step all of our work, all of our challenges, developments, and it will be shared with everyone who will be interested in joining us and contributing to the building of the biobank using both ex situ and in situ populations. But for today, we'll only focus on those two steps of the project. Okay, when we want to build a biobank, we need to collect some semen from the males. And the first challenge occurs when we need to catch a penguin. We are proud and we're very happy to have one of the biggest enclosures and the pools for the penguins and one of the biggest colonies, now over 100 individuals. But you can imagine that there is a problem, first of all, with identification. It's quite hard to identify specific male when you want to catch it. And then it's also quite difficult to separate them from the colony. It's quite easy when the penguin is close to its nest to kind of lure them in and then you can catch them. But imagine when a penguin is standing on the edge of the pool, you're approaching it and it jumps into the pool. So it's impossible to fish out the penguin from a pool that is 4.5 meters deep and has huge amounts of water. So this is also very challenging. We all love penguins, right? We think they are cute. But are they really? I don't know if many of you had experience working with African penguins. Their behavior is quite different from the ones that live in Antarctica because they get agitated quite easily and they can be very aggressive, especially during the breeding season. So believe me, African penguin will bite you, it will try to kill you. They are really strong, so it's very important, but also, on the other hand, challenging to immobilize them. You have to restrict them to make sure they are safe, sorry, but also to make sure that you are safe, and the person that is performing the dorso-abdominal massage is also safe. But when we finally get a semen sample, there is another challenge because the volumes are usually really small, between 1 and 50 microliters. And again, we're dealing with birds. That means that they're both reproductive and digestive systems and in cloaca. That is causing the contamination, which is quite often in this case. Okay, when we move to semen analysis and the challenges with that, we were lucky to have 42 males to sample from. That's why we got over 500 semen sampling attempts, but only 36% of those resulted in obtaining actual sample. But again, only 88 of them contained spermatozoa. And what you'll notice here, those numbers keep on dropping. So only 64 samples were ready for morphological smears, over 30 for CASA analysis, which are done for basic semen characteristics as concentration, motility, and these were due to contamination. Another constraint was low sperm count number, which only allowed us to run 18 flow cytometry analyses. And those analyses are quite important because they show us, they tell us about the DNA fragmentation in the sperm, they tell us about the sperm membrane, acrosomes, and many more factors. When analyzing semen, especially in birds, it's often that we pull the samples together. For example, if you would like to do some trials on roosters, you have a couple of roosters, you collect semen samples, pull them together, and then you work with this huge sample and you have a lot of material to work with. But in our case, we wanted to assess them individually, so we had a lot of samples, but again, they are smaller ones. So we've had lower individual rates, but on the other hand, we hope they were more representative for the population. And we've also managed to assess the morphology and morphological abnormalities, which have not been described for this species before. So if you would be interested in more in-detail information of our results, feel free to scan the QR code, which will direct you to the article. And here are some of our conclusions from this work. And from my perspective, I think one of the important findings that we had was that the level of habituation of penguins to human doesn't influence the outcomes of semen collection, which means that it doesn't matter if a penguin was only parent-reared or partially keeper-reared, the success in semen collection was the same. And for us, it's a good indication that in the future, if you would like to collect semen from the wild individuals, we will probably be able to do so. Okay, now moving to the cryopreservation part. I've already mentioned the small sample volume, because that was also the trade-off between assessment and the cryopreservation. So we weren't able to do everything in the one reproductive season. So firstly, we had to do the extensive semen assessment and then wait for another year, for another breeding season to do the cryopreservation trials. So here you can see different cryopreservation protocols for different species, just to show you that they need to be species specific. And that is mainly because of the spermatozoa, because it comes with various shapes and sizes for different species. And this affects how cryoprotectants work. And cryoprotectants are basically the substances that prevent spermatozoa from being damaged by ice formation during the freezing. And we have two types of those. So first are the ones that are permitting cryoprotectants, CPAs, that work from within the cell. They push out the water particles and protect the cell from the inside. And the DMF is one of the examples. And the trehalose is an example of external or non-permitting cryoprotectants that also cause cell dehydration, but they will protect the cell from outside. One of our big challenges was that we didn't know anything about the basic requirements for the ejaculate, because the ejaculates at the cryopreserved samples are intended for artificial insemination in the end. But there is no data on that, so it was quite hard for us to establish the basic requirements, but we had to go with something. So we just established that we'll go with a high total and progressive motility with low morphological abnormalities, and we've assumed that 1 million spermatozoa per ejaculates will do. It's always best to test as many cryoprotectants for the specific protocol to find out which one works better. And here you have some examples of how many there are. But that also would mean that the small sample volume would have to be divided in that many smaller ones. And it would be also great if we choose a cryoprotectant to test different concentrations of it. But that would also mean keep on dividing the samples into even smaller samples. So because of that, we have only chose those 2 cryoprotectants for our trials. And again, if you would be interested in more detailed results of our studies, you feel free to scan the QR code. Some of the results were that the DMF, the internal cryoprotectant, was better in preserving motility and mitochondrial potential, but on the other hand, the trehalose was better in preserving acrosomes and DNA integrity. And as for some take-home messages, it is quite challenging, mainly logistically and biologically, to establish a biobank for African penguins, but semen collection via dorso-abdominal message is achievable. And as I've mentioned a couple of times in the presentation, the small sample volume is the main limitation for the studies. But despite of that, the cryopreservation of this species semen is achievable. But on top of that, the cryoprotectants work in different ways depending on the semen parameter that is assessed. So our study kind of indicated that the optimal universal approach cannot be applied in our case. And for the building of biobank, when we want to preserve individual genetic diversity from all the males, it would be best to focus on so-called targeted cryopreservation. So that basically means that, for example, if we have a male with a low motility of his spermatozoa, it would be best to use higher concentrations of DMF. And on the other hand, if we would have a male which acrosomes or DNA is more fragmented, it would be best to use more trehalose in the cryoprotectant solution. So that is all for me. Thank you for your attention. You can always contact me via email unless we have some time. I'm happy to answer any questions. Thank you.
Thank you very much, Paweł. I'm also a person, by the way, who fell into love with penguins. And I can't, or better said, I don't want to believe that they can be evil, but...
Yes.
Okay, are there any questions?
Hey Paweł, there's an online question for you. Is it possible to train the penguins to have the semen taken, better for stress also?
Yes, so training birds for semen collection is quite usual for the captive environments. But part of our studies was not to do so. We have a colony that is quite big, so we have, let's say, 50 penguins, and half of them were parent-reared and half of them were keeper-reared. So when we are hand-rearing penguins, they are accustomed to people. So we accustom them from very early onset of their lives to not be stressed during handling, during the regular zoo procedures. So those ones are, let's say, trained for handling, but also you can train them for the semen collection. But in the back of our heads, there is this perspective that we would like to do the semen collection from the wild individuals. So it's impossible to train the wild individuals for semen collection. So to answer the question, yes, it's possible to train them, but the point of building a biobank when it would involve wild individuals, it's quite impossible. But we've achieved the results that shows us that they don't have to be trained, because some of our individuals, they're not accustomed to our keepers, but still they were like fighting us when we tried to collect the semen samples. But if they were in the right stage of the reproductive season, they still provided us with the samples that were really good for cryopreservation.
Just a quickie, Paweł, that's brilliant. My take home from that, it's more of a comment than a question, but it's just how difficult this is. It's really easy to talk about biobanking, but it's incredibly complex, and it just reinforces for me what a really good idea it would be not to lose the wild population in the first place.
Yeah.
And just, yeah, it's amazing stuff you're doing, and keep doing it, but it really brings it home.
Yeah, thank you. I was lucky to actually talk to some people in South Africa that work with penguins, and because of them being on the edge of being extinct, like some of the people like agree that, okay, biobanking is great, let's think about it, but on the other hand, don't approach the wild population because they are so precious and we don't want to disturb them. So there is also like a fine line between how we can do it. So we are now obviously focusing on the ex situ populations, but it would be great to preserve some, at least some of the wild individuals for the future. So yeah, okay, thank you. Oh, one more? Yeah.
I was too slow, probably. Thank you, Paweł, for this presentation. I also have a lot of experience with semen collection in mammalian species, and in my work I always try to connect it with reproductive behavior studies, and maybe we should talk later and see how we can optimize the semen collection procedure.
Definitely. We did, I didn't talk about it here, but we did also the extensive like reproductive behavior analysis prior to the start. So yeah, let's talk.
Okay, thanks.
Thank you.
Thank you very much. And now we will have a refreshment break. I think yes, for 10 minutes up to 11:45.